Hi everyone, thank you for joining me today. The title of this talk is A Framework for Brain Atlases, an overview and discussion on selecting an atlas for a study. Here are the learning objectives. After this presentation, my goal is for you to articulate the atlas concordance problem. What is it and why do we care about it? Then I want you to name the 4 common approaches in creating brain atlases. And last, hopefully you are able to question or examine the utility of any brain atlas for your study using a specific framework outlined here. I want to be realistic, however, in saying I don't have all the answers, but there are some important questions to consider near the end of the presentation and some key takeaways I hope you all can get here today. Here's the outline of my presentation. Of all the talks here today, this one falls into taking a broad overview of atlases from a 10,000-foot view. I first start with a discussion on the atlas concordance problem and the 4 common approaches in creating them, and even though some concepts here may be potentially mentioned in other talks, I think it is still helpful reiterating them in a different way. After taking a survey of many atlases, I will talk about some of the differences between them, including the wide variety of atlas features. Then I will tackle the question, does atlas choice matter? And finally, I will end with the framework for brain atlas selection and some conclusions taking approximately 25 minutes total. Now an overview of brain atlases. I think it is helpful to start with specific definitions so that we are all on the same page with concrete terminologies. So when I say brain atlas, I mean a neurological map that defines discrete brain region labels. They are discrete and not to be confused with brain templates, which are more continuous in nature. Now I think of brain templates like a cookie-cutter template where it outlines a representation of what a brain should look like. You can have templates of normal and abnormal T1, T2 images, but also of other imaging modalities like gray matter probability maps, PET, and DTI. I want to distill these 2 terms, brain atlases and brain templates, because they are sometimes used interchangeably in the literature of the last decade or are a combined term, but they are distinct things. And I also want to separate out the colloquial uses of these terms, atlases or maps, where we are indeed creating maps of brain features such as EEG characteristics across the brain. But these studies are not necessarily talking about discrete brain region labels of specifically neuroimaging atlases. And then finally, I want to mention a third term that may be still confused but distinct is the coordinate system of the brain, or the stereotactic space with XYZ coordinates, usually in a common MNI space, which is usually considered the successor to the Talairac space. Now with the definitions out of the way, let's talk about the atlas concordance problem. This problem was first defined in 2009 as an issue that stems from the difficulties and differences in the description of brain structures, which presents certain obstacles for the neuroscience research community. The term brain atlas has risen exponentially after the last 30 years, especially after the introduction of bold fMRI, where there was more of a need to map the brain to relate findings across studies, and many atlases have risen since this time. But I want to say, don't feel overwhelmed by the number of atlases, even as someone who has spent time looking for all the atlases out there, I still hear of new ones. Here, I try to summarize the more common ones and many features, particularly in this table from our lab's paper on questioning if atlas choice matters. And the supplemental section includes more complete tables outlining some of the history and creation of each atlas, along with their sources. But I'm not the first to create such tables, and it is worth mentioning again that there are so many atlases and their variations that these tables include atlases not in mine, and I have atlases not in theirs. And so the atlas concordance problem states that we currently lack a thorough understanding of if atlas choice matters for a study and the potential challenges that discordant parcellations pose, particularly for meta-analyses and other information integrating efforts. Just like in creating many atlases of the brain, we can have many atlases of the world at different scales depending on our needs. But different atlases will call the same structures different things. For example, the AAL atlas shown here will label the superior temporal gyrus with the superior temporal pole and the superior temporal regions. The Cerebra atlas combines these two regions as just the superior temporal region. Now the Hammersmith atlas includes the posterior superior temporal gyrus, but the Harvard-Oxford atlas will include an anterior superior temporal gyrus, where 90% of the voxels in this region will overlap with the posterior superior temporal gyrus in the Hammersmith atlas. Now this can be problematic, as I mentioned before, for meta-analyses summarizing findings across studies where the labeling themselves can be antagonistic. However, the specific labels of each region may not necessarily matter for some studies. We can call these regions anything we want. In the analogy of creating atlases of the world where names can change, but geographic labels essentially represent the same regions, this analogy falls apart in that, while there is one Earth, there are many brains. Essentially, we are trying to create one atlas of the brain that is conserved across all individuals and potentially across species. We are essentially creating one atlas for an individual that does not necessarily exist by taking averages across multiple individuals. And this line of thinking, that there may be not one single atlas for all modalities, for all individuals at all given time points, is gaining traction. Next, talking about different atlas types, they can fall into one of four categories. In the table here, I have listed S for structural atlases, F for functional atlases, M for multimodal atlases that were created using data from multiple modalities, and V for variable atlases where they can be structural or functional. This includes random atlases where the regions can be entirely random, or you can have random subparcellations of structural or functional atlases. Another dimension to consider in atlas creation is the fact that atlases can be registered to individual brains using surface-based algorithms or volumetric-based algorithms. Surface-based registration can provide superior labeling of cortical regions over volumetric registration, but for other atlases where their regions extend deeper into the brain, like the AAL atlas, registration methods may not matter as much. So you can see here that there are many atlas features across the literature. You can have structural, functional, multimodal, volumetric, and surface-based atlases like I mentioned before. We can also have atlases based off of connectivity data, atlases based off of cytoarchitecture, molecular, and genetic data. You can have atlas regions that tend to the sizes of voxels with thousands of regions, or tend to the size of loads with very few regions. Now, some atlases only include cortical structures, while other atlases include both cortical and subcortical gray matter structures termed gray ordinate, but still leave out white matter regions. Thus, there are also white matter atlases. Now, sometimes in the literature, you will see investigators combine atlases because none of the available atlases meet the needs of a study. For example, if they need both gray and white matter regions. Some atlases will have symmetrical labels, like the Harvard-Oxford atlas, that you will need to divide in half if you want to separate out left versus right sides of the brain. And some atlases will include the entire brain, like the Talairach atlas shown here. Worth mentioning are the efforts to map the brain across different species and other efforts to map very specific regions into very fine detail like the cerebellum or hippocampus. Some studies may employ so-called study-specific atlases where regions are created from labels of the very participants from the study itself. Other atlases will liberally extend deeper into brain matter rather than follow the cortical surface conservatively. Rarely used in the literature are soft atlases, where region labels can overlap with other regions, on contrary to hard atlases where region labels are considered very discrete. Next, atlases can be derived from other atlases like the AAL 600 atlas, which is a further parcellation of the AAL atlas. And finally, atlases can be completely random. Now, there are many atlases with non-mutually exclusive atlas features shown here, and I'm sure there are many more categories to come to meet the needs of the investigators. Next, I want to briefly go over some of the physical differences between these atlases with a focus on their morphologies such as size and shape, and structural connectivity networks created from these atlases to give you guys a sense of what these atlases may look like in comparison to each other. Here you can see 5 different atlases and the distribution of the sizes of all their parcellations. On the x-axis of the density plot, you have the volume of parcellations on a log scale, showing that some atlases have a very uniform parcellation size, such as acrotic atlases, which were created with the intention to be more uniform, and other atlases that vary widely in their sizes, such as the AAL atlas. Looking at another morphological property, such as sphericity, where a value of 1 represents a perfect sphere, you have some parcellations that are more spherical and great for some study designs, and other regions that may be more ellipsoid. Combining both sphericity and volume distributions of these so-called standard atlases as opposed to random atlases shown next, you do have regions that are very large and non-spherical in the bottom right-hand side of the graph, and other regions that are very small and spherical on the top left-hand side of the graph. Now, in comparison to standard atlases, you have atlases composed of completely random parcellations. The benefit of these atlases is that you can vary the sizes and shapes of all their regions or keep them uniform. You can create multiple iterations of these atlases, which allows you to repeat results across many atlases to see if the results do hold up. But the downside to these atlases is that the regions are random. In other words, they don't represent any anatomical or functional entities other than following the outline of the brain mask that you use to create them. Now, I know that many people that use these atlases are in the network world and create connectivity matrices based off an atlas. What I wanted to know is how connectivity, specifically structural connectivity seen here created from diffusion imaging, changes across parcellation scales, which was shown in the now classic paper by Zaleski et al. Back in 2010, but also how does structural connectivity change across different atlases commonly used in the literature. And so we can visually see here that yes, networks do look different across different atlases at different thresholds. And so I looked at some simple network features such as density, mean degree, and clustering coefficient, and see how these features change at different parcellation scale in both random and standard atlases. But so what? We see that these atlases look different, created for different purposes, have different features, make different networks, but does it all really matter? Do I need to use functional atlases for functional data? Do I need to use structural atlases to create structural connectivity data? Can I use completely random parcellations? Can I leave out entire brain regions like the cerebellum? Now, these are all questions I had when I first began my PhD many years ago, because my goal was to use these atlases to help answer a clinical problem. I'm not necessarily here to create atlases or be a so-called brain cartographer, but I'm here to use these maps to guide me through the landscape of the brain to find seizure onset, which brings me to my next section. I want to go over briefly our study mentioned before on using these atlases to be able to test hypotheses about epilepsy pathophysiology. Now, I don't have time to go over the entire study, as explaining a structure-function analysis employed in the study with justice can take me a good 5 to 10 minutes in itself, but I think it's still helpful to go over some of the background along with some of the main results to answer the question, does atlas choice matter for this particular study? Shown here is the implantation of stereo electroencephalography, or SEEG electrodes, to localize seizure onset more precisely for surgery. Now, the problem with this implantation modality is that we have a sampling bias in that we cannot implant all regions of the brain. And thus, where we think seizures are coming from may actually be coming from regions where we have no implantation. Now, how do we solve this problem? Well, that's where whole brain neuroimaging can come in, where we do use structural data to find epileptic functional activity, but it's still region-based. We need an atlas using network methods to answer questions such as, what parts of the brain should we resect to cure our patient of epilepsy? Now, you can imagine with very large parcellations, our answer could be biased and say to excise an entire superior temporal gyrus, whereas other atlases may say that we need to excise a smaller portion. Now, in our study specifically, we looked at how the structure-function relationship changes at seizure onset, which has been explored previously in other studies using very similar methodologies. What I wanted to know was, how does atlas choice affect our results? Does it affect our effect sizes, in other words, our power? And fortunately, the answer was no. Many atlases gave us the same answer, although with different power or coens d. So, we can use random atlases, we can use functional atlases to create structural connectivity networks. And generally, trends that we did see was at very gross parcellation sizes or very, very small parcellation sizes, we tended to see no differences or small effect sizes to test our hypotheses. And other atlases with very different structural parcellation protocols, such as the Hammersmith atlas, gave us outliers at certain parcellation scales. And so that brings me to the framework of selecting an atlas for a study and some of the lessons I learned in conducting this study, broken into different parts. The first part is more of a theoretical framework that generalizes atlas choice in alignment with a specific goal and with respect to some atlas features. If the theoretical framework is too theoretical, which we did get criticisms during the peer review process, we also have some practical questions to consider when thinking about atlas selection, and may save you some time early on. Finally, the last part of the framework discusses some guidance about what to do before and after your study, specifically relating to what are your goals in using an atlas and what happens when you get conflicting results. Now, when discussing the theoretical framework of choosing or creating the best atlas for a study, there are parallels in creating appropriate models, such as network models or animal models, in that you want to maximize three different things. These three things are descriptive validity, explanatory validity, and predictive validity. Descriptive validity is exactly as it sounds. The atlas matches or describes the system in which you work. It contains the atlas features that you are interested in. Obviously, if you want to explore white matter structures, you need an atlas that contains white matter. But a little bit harder to conceptualize or know what the answer to is selecting an atlas at the appropriate scale based off of the measurement modality. For instance, you don't want to select an atlas with very, very small parcellations in which your measurement modality incorporates signals over large volumes of space. In the case of scalp EEG, where sometimes you need pathologies as big as a fist to see any changes on EEG, atlases with thousands of parcellations may be inappropriate to localize seizure onset with this measurement modality. Similarly, for intracranial EEG shown here, which I use, we are assigning specific signals captured on intracranial EEG to specific regions, and having many parcellations in an area of the brain may falsely attribute a signal characteristic to more regions. Now, I'm not an expert in fMRI, but similar principles hold up to see how we are assigning specific metabolic changes to large or small regions of the brain based off of imaging resolution and other parameters. On the other hand, having too large of a region may drown out our signal from the noise, and thus we have lower power to detect interesting findings. Is that we want to maximize either explanatory validity or predictive validity. This relates to our particular goal in conducting our study and is based off of a classical statistical paper back in 2010 to explain or predict. This paper discusses different goals to explain or predict, and the main thesis of the work is that clearing the current ambiguity between the two is critical not only in proper statistical modeling, but more importantly for proper scientific usage. Both explanation and prediction are necessary for generating and testing hypotheses, yet each plays a different role in doing so. The lack of a clear distinction within statistics has created a lack of understanding in many disciplines of the differences between building sound explanatory models versus creating powerful predictive models. And one thing the paper reasoned was that a good explanatory model may not necessarily be good at predicting. Similarly, I think there are good parallels in selecting an atlas for a study. For example, If we're using an atlas to test or explain a biological phenomena of a very specific region in the brain, such as a thalamic subnuclei, you better be sure that the atlas correctly localizes and assigns the signal or measurement characteristics to that region. But on the other hand, if you are into predictive modeling, such as our lab, and predicting how does the structure-function relationship within the brain changes at seizure onset, or if a patient is going to be a good or poor outcome after epilepsy surgery based off of structural network data, then having very accurate parcellations may not necessarily be your goal. You want a pipeline that, at the end of the day, works for our patients. If iterating results over many thousands of random atlases increases our confidence in our prediction, even though the regions themselves do not represent specific regions, then that may be an appropriate atlas selection. So the reason why I don't think anyone has or ever will have a direct answer to which atlas should I use, the answer is always, as many times in science, it depends. Now, to some, a theoretical framework of descriptive, explanatory, and predictive validity may be too abstract, so we do have a list of practical questions to consider outlined in blue at the bottom. Some of these questions I have hinted at throughout this presentation, but the main ones are: do I need accurate parcellations? Can I get away with using random atlases? Another important question to consider is if I use a custom atlas, such as one that you made yourself or subdivided or combine different atlases together in your study, how will that affect the replicability of my study or the communication of results to other investigators, such as an increase in X is found in the anterior superior temporal gyrus, like I mentioned at the beginning of this presentation. Another question is, are there any consequences from leaving out brain regions such as white matter, cerebellum, or other subcortical structures? And another final important question is, what are the computational costs, resources, and time and personal training needed to use atlases, even such as newer atlases using deep learning models? Some atlases may not have the computational, resources or the personnel or training to deploy such atlases. Finally, the last part of this framework discusses what to do before and after a study. Before a study, you may want to consider everything in the framework I discussed before, including the practical questions, atlas features, and each of the descriptive, explanatory, and predictive validities. Another thing you may want to consider is selecting a standard set of atlases across scales and created with similar or different features. Now here, you have to balance power because the more tests you run across more atlases, the lower your power, but if there are repeatable results, then that leads to more robust conclusions in your own work. Now, similarly, for people creating tools and other software, it might be beneficial for all of us to get together and have a discussion on deploying a minimal standard set of atlases incorporated into pipelines. Now, our own lab has encountered problems where we may want to use a public brain network dataset, but the published data does not contain fully the regions we are interested in. Now, finally, in this third row here, the last part I think is the hardest scenario: what to do if conflicting results between atlases arises. Now, fortunately, in our study on the structure-function relationship in the brain during seizures, we either had enough power or not enough power to detect differences. This is in contrast to the potential scenario where prediction reverses directions or other finding reverses directions. I think the best way for now to go about this is that we need a better understanding of the biases introduced because of atlas parcellations such as scale. Now on to conclusions. With all of that being said, I think one solution I am currently employing in localizing seizure onset is using a consensus across multiple atlases. So here I take many standard atlases and random atlases, find their regions of prediction for most likely seizure onset, and average their findings to localize seizure onset. That is one method. But largely, I consider this to be a band-aid on the whole problem. So where do we go from here? Well, ideally, we want to find the true map of the brain that represents the true organization of the brain at the top of this diagram here, where the bottom level, the first level, represents the creation of brain maps. The second level represents our understanding of when do and don't atlases work for specific scenarios. And finally, the top level, the third level, is the creation of one or multiple brain maps that represent the true underlying organization of the brain. Like I said before, there may be never one single true brain map for all scenarios. Potentially, we can have one multi-dimensional atlas, including multiple modalities, which we can vary across scales using a sliding scale. But currently, I think we are between levels 1 and 2. And with that, thank you for listening to this talk. I hope it gave you some satisfying or practical answers to your questions with a healthy dose that there's still lots to do.